We started exploring these venoms in the mid-90s, and I like you, thought, why does a spider need more than a couple of insecticidal compounds to kill its prey? Surely. I was absolutely shocked when I separated the venom for the first time and saw that there were literally hundreds of components in there.
Fellow Homo sapiens, my name is Torie Robinson, and welcome back to Epilepsy Sparks Insights. Now, who relates spiders to epilepsy? It turns out that spiders, amongst some other cool organisms, can contain amazing compounds that could help stop seizures in people with refractory epilepsy. Today we hear all about it from the cool biochemist, lead researcher and PI, Glenn King.
Hi Torie, lovely to be here. My name's Glenn King and I'm a professor in the Institute for Molecular Bioscience at the University of Queensland. So it's a research institute of the university.
And what do you actually research?
We're interested in human diseases, what I'd broadly call neurological diseases, where the underlying defect is in this receptor called an ion channel. These are things that sit on the outside of your nerve cells and let ions like sodium, potassium, calcium across. Many diseases are actually caused by defects in those ion channels. And of course, one of those diseases is epilepsy. But our unusual take on this is that we use animal venoms as a source of compounds to correct those defects. And you may say, well, why animal venoms? Turns out that after 20 years of researching on animal venoms, we realised what they're full of, particularly invertebrate venoms, things like spiders, scorpions, assassin bugs, centipedes. They're full of molecules, little proteins called peptides, that actually modulate the activity of ion channels, the very things we're interested in. In fact, they're the best known source on the planet of things that will modulate the activity of epilepsy ion channels.
Of any other species that we know of, are these guys? These are the creatures that do it?
Absolutely. The creatures that do it. And the cool thing is, they're some of the oldest creatures on the planet.
I was just thinking that.
Yeah, they've been around a long time. Like spiders, centipedes have been around for 400 million years. So they've been actually working on the problem of making these ion channel modulators for a heck of a long time. And so they've tuned them up really nicely for us.
How does this product, how does it benefit these species? Like, why do they have it?
Yeah, great question, Torie. Yeah. So a lot of these things are actually either will paralyze or kill insects. So they have the same ion channels as we do, or closely related ones, but they use them in different ways. So something that might kill an insect might actually be beneficial for us. So they have a purpose for them, but we're repurposing them for something different.
So this is something brilliant to come out of the evolution of these species because it's of benefit to us.
Absolutely. Look, there's a classic example. There's a drug at the moment that comes from a deadly cone snail. So the cone snails are these little venomous cones, slow-moving mollusks that live at the bottom of the ocean. And believe it or not, they catch fish. And the way they catch fish is they have venom, and they actually, this sounds like science fiction, but it's true, they load it into a harpoon and they actually shoot it at the fish to paralyze the fish. Can you believe that?
That is so cool.
And the cool thing is, in that venom of that cone snail, there's a toxin it's a toxin in the ecological context that actually paralyzes the fish by paralyzing their neuromuscular junctions. Now, we have the same channel, but in us, it's in the spinal cord and it's used for transmitting pain to the brain. That same molecule's now been developed as an analgesic for humans because it's a painkiller in humans, but deadly to a fish.
That's amazing. So we're getting stuff from a snail, for example, that is benefiting us as we're all related somehow, right? But just like that, there's that distance between us, yet it is...
It's huge. Yeah.
Which type of spider is it that you are working on or with?
We're pretty agnostic about the source of the ion channel modulator. So we have a collection of more than 500 different venoms from different species of arthropods around the world. So there's, as I said, there'll be spiders, scorpions, assassin bugs, caterpillars, bees, wasps, ants, whatever. And when we have an ion channel that we want to target, we'll screen our venom collection against that ion channel, looking for a venom that might have the activity we're looking for. The hard part is when we have a venom that we know has something in it that has the right activity, we've got to find that thing, that compound in the venom. And some of these venoms can have hundreds to thousands of compounds in them. So now we've got to do a deep dive and say, which is the molecule that has the activity that we're really interested in, and that's where most of the hard work is.
Gosh, yes, I just have to say I feel so ignorant. You know, the more you learn, the more you realise you don't know. And I was, you say, oh, I have to go through all these different compounds within the venom. And I was thinking, yeah, I'd never even thought about that. That's, a lot of work. I mean, how many different compounds or things within a venom may there be for you to have to identify that one thing?
I mean, to give you an extreme example, the spider that we've worked with most is the Australian funnel-web spider, particularly one found on Gari, which is the biggest sand island in the world, just north of Brisbane. And that spider has more than 3,000 peptides in its venom. So when you're looking for a molecule that has a particular activity, you've got a lot of work to do to narrow it down to find the right compound.
Wow.
And look, I was the same as you. When I started working on venoms, my interest in venoms initially was to try and develop more eco-friendly insecticides. And I thought, well, spiders are the best insect killers on the planet. Surely they've worked out the chemistry and pharmacology of making insecticides a long time ago. And so we started exploring these venoms in the mid-90s. And I like you, thought, oh, why does a spider need more than a couple of insecticidal compounds to kill its prey? Surely, doesn't at most maybe. So I was absolutely shocked when I separated the venom for the first time and saw that there were literally hundreds of components in there.
It sounds like really impressive and quite humbling, really, just to hear you talk about it and the diversity of these venoms.
Yeah, they're amazing, absolutely amazing. And even the latest venomous animal we've been looking at is caterpillars. There are actually quite a lot of venomous caterpillars. So they're venomous at the caterpillar stage because they're very, very sedentary and very easy to be preyed upon. They're not venomous as adults, though. So when they turn into moths and butterflies, they're not venomous anymore. And very few of them have had their venom looked at. And it turns out they've got really interesting and complex venoms. And so we're adding those to our venom collection as well now.
So these venoms, you've been testing them, I believe, in upon epilepsy in rodents. Is that correct?
In the past, that's right. We would have a receptor that we're interested in, an ion channel. We'd screen our venoms. We'd find the venom that was working. Then we'd isolate the component from that venom that did the job. And so this was all in vitro work, just testing it directly against the channel. And then you're right. Then once we got something that we thought was good, then we tested in a rodent model of the epilepsy.
And what were the results of that?
So the original one we worked on was Dravet syndrome, which, as you probably know, is a deficiency in a voltage-gated ion channel called NAV 1.1. And we were looking, so patients have less of that than they should. And so we're looking for something that would just spice up the activity of the remaining amount of NAV 1.1, what we call an agonist in pharmacological terms. And we found this beautiful, very selective molecule that just potentiated the activity of that channel. And so we tried that in a mouse model of Dravet. It's a very severe Dravet model in the genetic background we have it in. And so the mice start to seizure at about postnatal day 17. It gets pretty bad by about 19, and they rarely last more than a week. It's a very, very severe form of Dravet. So we started infusing this peptide from a tarantula into the brains of these mice at day 19. Within 4 days, we'd eradicated the seizures completely, and the mice survived, unlike the control mice who all died within a week. So we're able to pretty much completely eradicate seizures in that model of Dravet.
Were there any side effects to treating them with this venom?
Well, we only did it for about a week or so we don't know whether there's anything long-term. And this is where we ran into trouble because this was done just prior to COVID. We decided we needed a molecule that had a more long-lasting effect. We didn't think the effect from that was going to be long enough clinically. So we developed a better molecule. And when it came to thinking about testing that in that rodent model, COVID happened. And, of course, we could not get access to animals during that period of time. And that's when we started thinking about perhaps a better way of doing it.
Tell us more. So your better way of doing it?
I mean, the problem with the rodent models of epilepsy is, as you know, that for each particular type of epilepsy, I guess we should pull back a bit and say, for people who aren't used to talking about epilepsy, there are many, many different types of epilepsy. And even within a class of epilepsy, say Dravet, There are many hundreds or even thousands of different mutations that can lead to that class of epilepsy. And so when you test a rodent, when you use a rodent model, you're testing just one mutation from that patient group. So you know it might work for that mutation, but maybe it's not going to work for all patients. And so what we can do now is take advantage of the revolution in stem cell biology. And we can take blood samples from individual epilepsy patients, and from them we can derive a brain organoid, a little mini brain, just a few millimeters, that contains all the cells that are in the human brain that talk to one another in the way that cells talk to one another in a human brain. But the good thing is we can make those organoids from lots of different epilepsy patients, all with the same nominal epilepsy, but with different underlying mutations. And then we can actually take this little mini brain and we can put it on something called a multi-electrode array. All it is something that measures the electrical activity of that brain. And we can see for the epilepsy patients, it's a bit different. It's overexcitable. And then we can just put our candidate drug on there and say, does it correct the phenotype in that mini brain from that patient? And if it does, we can say, well, that's great. But what about the 5 other patients that have that epilepsy with different mutations? Does it correct the phenotype for those ones as well? So we can look at a broad array of patient mutations now and develop more what we would like to call personalised medicines that we know are going to work for that particular epilepsy. So I should make that point. We're not interested in making generic epilepsy drugs. We're interested in making personalised epilepsy drugs for specific types of epilepsy.
But I have to say, as a person with an epilepsy, that sounds beautiful to me. Having a drug that is specifically, well, basically designed, literally, right,
Correct.
for the individual. So,
yes.
And I presume with that, it's likely to be, one would hope, more effective in seizure control, but also, I imagine, like side effects, you'd be looking at those to minimise those for people as well, right?
Correct, yeah. So we're working with peptides, not small molecules. So small molecules tend to be easy to make and permeable to the brain, so they're good from that point of view. But because they're small, they often bind to lots of different things because they can fit into lots of crevices in all sorts of receptors. So that's where you get the side effects. The peptides we're working with are bigger, and they bind to a greater surface on the receptor, and therefore they tend to be much more specific, usually more potent, and also much more specific, so you don't get the side effects that you were talking about. That's for sure. Or at least less likely to.
So you've got your organoids.
Yes.
Lots of people will say, especially if they're not so familiar with research, okay, cool, how long before now with the organoids, and assuming, like, of course it's going to be successful before it gets to humans. So could you just break that down for us a little bit and tell us where we are in the grand scheme of things, please? Yeah,
look, it's still a long way to go. So what we have, in the case of Dravet, for example, we have a drug that we know works in the animal models and it also works in the organoids. But then you have to do a whole bunch of safety testing, of course, before you even go into clinical trials. And then there are 3 stages of clinical trials, so-called phase 1, 2, and 3. Phase 1 is just to look at the safety of the molecule in usually healthy volunteers, not necessarily epilepsy patients, and that alone can take a year. And then you've got the 2 phases where you're looking for efficacy, phase 2 and then a much bigger phase 3. And that can take up to 3 to 5 years. So the whole process from discovery to having a drug is 10 years plus typically.
And not all of them make it, right?
Absolutely.
However, I always say, though, we do learn a lot about diseases in the process, and that information can be used in other studies or to develop an idea further.
Absolutely. No question about that. You learn from, they're not necessarily errors. It may be that the pharmacology of the rodent doesn't translate fully to humans, right? So your drug might work brilliantly in a rodent. I mean, cancer has been cured in rodents so many times. But that doesn't always translate to humans, unfortunately, and that's the risk you always take.
We are such frustrating organisms, aren't we? The overcomplexity does my head in, excuse the pun. Thank you so very much to Glenn for making epilepsy research cool and relatable, and for sharing with us his work using both rodents and organoids. Also, if the term organoids rings a bell, that might be because a few weeks back we also chatted with the fab clinician and researcher Patrick Kwan, who is working with Glenn. So check out that episode here if you haven't already. You can find out more about Glenn and his work on the website torierobinson.com, where you can also access the podcast, the video and the transcription of this episode. And if you haven't already, don't forget to like, comment and subscribe to the channel. Share this episode with your friends, colleagues, family members, and see you next week.